everybody, and welcome to Lecture 4 for Human Biology. This is going to be on the muscular system, so let's dive right in. All right, what I've got here is a quick breakdown of the major details, structure and functions, if you will, of the muscular system. Just some general information that is worthy of your knowledge. For instance, realizing that your body is going to have a large percentage of its weight built up into musculature, so your musculature makes up about half your body's weight overall. That's a huge amount for a single system. You have lots of individual muscles, somewhere in the neighborhood of about 600. The concept here is if you got a little over 200 named bones, you'd expect those bones to do multiple things, and for those bones to move around based off the fulcrum-like action of your joints, you must have more muscles than you do bones. Again, large percentage of your body weight. This comes in 3 flavors. In your system, you have skeletal, cardiac, and smooth muscle, and they are all different from one another in ways that allow them to do the jobs that they are responsible for. So they are quite different, in fact, leading to their general function. Skeletal muscle being what you think of as the muscle you use on a daily basis. Cardiac muscle being that which is involved in the heart, and smooth muscle really all over the body, but primarily you think of as being part and parcel of the intestinal tract. All of these are going to be specialized for one major purpose. That purpose is, quite simplistically, to take chemical energy in the form of ATP and turn that into motion, okay, to move. So what your muscles do, their goal at the end of the day is to take ATP, chemical energy, and turn that into motion, right? That is how your muscles do business. Now, again, 3 types of muscle that are worthy of your consideration. These are skeletal, cardiac, and smooth muscle, and they are set up for the jobs that they do. Skeletal muscle, very important because it is both striated and voluntary. Striated and voluntary. It is striated, it has a striped appearance, and it is voluntary in that you can consciously control it. I think flex bicep, and my bicep flexes, that's skeletal muscle. Skeletal muscle is going to be good for very powerful contractions, but it also tires readily, all right? So if you hold something and you hold it straight out, you're not gonna be able to hold it out for very long because your skeletal muscle tires quickly. This is sort of in the setup of it in essence. Cardiac muscle, on the other hand, is also striated in its appearance. It also has striations, but it is involuntary. So your heart beats on its own. You don't have to think beat heart every time you want your heart to beat, or else you'd be doing that all day long. So it is striated in its appearance, but it is involuntary. And it's very different in a lot of other ways from skeletal muscle. For instance, skeletal muscle cells tend to be single, big, long cells. Like you'll have one cell that runs the length of your bicep, whereas cardiac muscle cells are quite small. They are very, very tiny. They're just sort of bound together. Okay, so instead of having one big long cell, you'll have a bunch of individual cells. So skeletal muscle, long cell, cardiac muscle, individual cells. And then, of course, there is smooth muscle. Now, smooth muscle is quite different compared to the others. Instead of having the traditional kind of squarish shape that you'd think of, smooth muscle cells have a shape where they're wide in the center and then they taper off to tips, all right? You can kind of see this down here in this image. They have these unique shapes to them, and they are non-striated and they are involuntary. Now, the effect of striations are caused by what are called the myofilaments that you'll see soon, lining up and forming these distinctive bands. And when you have striated muscle, that muscle is capable of contracting quickly and strongly. Okay? Cardiac muscle and skeletal muscle, they can do this. They're striated. Whereas smooth muscle is not striated. When it contracts slowly and what you might call smoothly. Okay, slow and smooth, that is smooth muscle. Non-striated, involuntary. You don't think about contracting your smooth muscle. And smooth muscle is important because it also takes part in what is referred to as peristalsis. All right, peristalsis is a set of rhythmic contractions seen in the intestinal tract and all over the body for that matter. You see this in the ureters, you see this in the esophagus, you see this all over. But generally speaking, think about your intestines. What will happen is you'll close off one spot and then force everything down a tube, okay? It's a set of rhythmic contractions, closing and then pushing to propel things down the length of a tube, like your intestinal tract, like your ureters, etc., etc. All right. Now, General functions of musculature: obviously produce movement, maintain posture, stabilizing joints. These are all important concepts. But another very important bit to say here is that they generate heat. If you go and you participate in some sort of sport, if you decide you're going to go play basketball for an hour, you'll get hot very quickly because the physical exertion, the usage of your muscles, generates copious quantities of heat. The reason you get hot, the reason you get sweat when you're participating in some sort of sporting event, is because your muscles generate large amounts of heat just through their activities. Okay. Connective tissue of muscle. Now, there are several of these that are worthy of our consideration, but what you need to think about is that they are part and parcel of each other. Okay, they are sort of all working together in the same goal. It's just because they are found in different places, we give them different names. So, what are they? These are fascia, epimysium, paramysium, and endomysium. That's some weird color patterning there. Never seen that before. All right, fascia, epimysium, paramysium, and endomysium. Let me explain what this is. All right, if you take the muscle and you chop it down the middle, what you see in cross section is that the muscle is divided up into parts. These parts of the muscle are referred to as being fascicles, muscle fascicles. Fascicle, 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 fascicle. Inside each of those fascicles, you find individual muscle cells. Now, the outer covering of a muscle or a whole muscular group, like your whole quads, all right? All of your quads and your leg, they will be surrounded by one big thick layer of fibrous connective tissue, and that big thick layer of fibrous connective tissue on the outside of whole muscle groups is called fascia. Next, in each individual muscle, every muscle in your body, they will all have a covering of what's referred to as epimysium. Epi means upper, mysium, myo, anything my is muscle. Epimysium, that's upper muscle covering, okay? So that's a covering on the outside. If you look at the individual muscle itself, the whole muscle is going to have an epimysial covering. Next, we have paramysium. Paramysium surrounds the fascicles. Okay, so we've got our fascicle here. Paramysium surrounds each individual fascicle. Now, the paramysium is continuous with the epimysium. So the epimysium, the paramysium, and the endomysium, they are all continuous with each other. Okay, so they are not completely separate units. It's all kind of the same stuff. We just give it a name based off of where it is in the muscle. So epimysium's on the outside, paramysium is around the fascicles, and you guessed it, endomysium is around the individual muscle cells. So the individual muscle cells all have an endomysium covering. And the reasoning for this is pretty darn straightforward from my perspective. The middle of the muscle has a lot of muscle and not as much connective tissue. And then as you tail towards the ends, the muscle gets thinner and thinner, but the amount of connective tissue remains the same. And by the time you get to the ends of the muscle, there's like no muscle left. There's nothing but the connective tissue that runs all the way through endomysium, paramysium, epimysium, you name it. And that becomes the tendons that attach the muscle to bone. So this fiber, all this fiber inside of the muscle, it's like a structural support unit internally. It helps to hold the muscle parts in place and to give it structural strength. And as it tails off towards the ends of the muscle, you end up making tendons out of this same material. And further, if you think back to our histological conversations, you know that connective tissue, which is what this is, epimysium, paramysium, endomysium, it's just connective tissue. You know that connective tissue bears lots of blood vessels in some cases. This connective tissue brings in huge amounts of blood flow to your muscles. Your muscles must have copious blood flow to be functional. Don't believe me? Sit in a chair in a weird way for 5 minutes and stand up and feel like one leg's asleep or something ridiculous, all right? Fall asleep on your arm and wake up and it's completely numb and useless. You have to have copious blood flow to keep your muscles functioning. And if you look inside of the muscle, this connective tissue bears massive quantities of blood to and from your musculature. All right, muscle attachment. So muscles attach in two ways. We refer to this as being indirect or direct muscular attachment. This is pretty darn straightforward from my perspective. Indirect attachment means tendons. Direct attachment means no tendons. Okay, no tendons. Do I have an arm? And we're back, and we have a friend with us now. What you see here, let's see if the wrist looks like. What you see here on the wrist of this critter is you can see all of these tendons, The muscles that run the fingers are actually way back here. You can see it. So I can run my fingers, you can see the muscles back here functioning. What's going on here is, this is an indirect form of attachment. You got the muscle, and then you have a tendon that goes to wherever the muscle's going to do work. That is an indirect form of attachment. And then by comparison to this, if you look back here, you can see the muscles attaching up here on the arm, and then coming back and attaching to the scapula. But where they attach to the scapula, there is no tendon. The glutes are the same process. Look at the rear end on this guy. All right, they don't have any form of tendon, no tendons of any fashion. This is direct attachment. The muscle fibers embed themselves into the bone directly. There is no tendon between the muscle and the bone. The muscle fibers terminate into the bone and connect to it in that fashion. That is a direct or fleshy attachment. Yeah, that'll work. There is so much that we could talk about there. You just don't even know. A lot of neat stuff. But let's just keep on rolling. Again, our goal is to keep these lectures concise. All right. How do skeletal muscles work? Well, there's a few terms to discuss here. In any motion that your body completes, there will be what are called prime movers. Prime movers do the job. Synergist means together, to help, together. Synergists are the helpers, okay? They assist with whatever the prime mover is doing. Antagonists work against the prime mover, and fixators hold things in place. So you can imagine, here I am holding up my drink can. This is one of those NOS energy drinks, all right? Here I am holding it up. I can lift it like so. And when I do this, the prime mover that does the job is my bicep, all right? It's the main muscle running the show here. That would be my prime mover, my bicep. A synergist would help. They are helpers. That would be like my brachialis muscle out here on the arm. So you could take your hand and put it against the side of your arm and lift something up, and you can feel the muscles in there contracting. It's not just the bicep. There's more in there doing work. They are helping. They are synergists. And then there are antagonists. So antagonists, this is kind of counterintuitive, but you can imagine that while my bicep is flexing to lift my arm up, that my tricep is also tense. It's not pulling per se, but it's tense. And what that does is, by having that antagonist there, is it lets me have control of my arm. I'm not just throwing my arm up using my bicep. I'm very controlled in the motion of my arm because my tricep is sort of keeping tension against the other muscle so that it can move with great control. So antagonists are important. They prevent excessive motion, let's say. Jerky motions you might get at. And then, of course, there are fixators. What you may not grasp, folks, is that in a skeleton, like so, I mean, not dump the scene on the ground, your arm isn't very well connected to the rest of your skeleton. In fact, if you look at this, you've got the humerus and you've got your scapula, and there is nothing holding these to the body except musculature. The only point of connection to the rest of your skeleton is way up here at your clavicle, okay, your collarbone. When you lift something up, all right, when you lift something up, there is very little support for that arm, very little support indeed. So what you have to have is a pile of muscle. So when I'm holding this thing up, you have a pile of muscle in your shoulder, and what it will do is it will go and lock everything in place. All right, hang on. I think we still have our fellow over here. What this is going to do is, when this arm is being held up in this fashion, all of this muscle, there's these big old sheets of muscle back here, like big sheets of muscle. And when the arm is lifted, they go and lock everything down. All right, they lock it down so that it can't move. It's all muscle holding the scapula in place. That's why the scapula has this large appearance to it, is because it's just covered in muscle, all attached back to the spine. So it's going to lock down like this. And by doing so, it fixes the arm in place prevents excessive movement, and allows for the prime mover to do the job that the prime mover needs to do. And that's how this goes, folks. The fixator locks things down. Now, worthy of mention here are two more terms. These terms are origin and insertion. Every muscle has an origin or an origin and an insertion. Some have multiple. But the idea is that the origin doesn't move when the muscle flexes, and the insertion does. Okay, the origin is non-moving, the insertion point is moving. So think about my bicep. The origin is up here. When I flex my bicep, I can feel the muscle moving right there. I feel it, but it's not changing location. Whereas the insertion point for my bicep is way down here on my lower arm. And whenever I move my bicep, it does move. It's got motion to it, okay? So the origin point is non-moving. It's where the muscle begins, and the insertion point is where the muscle terminates, and it is modal. The insertion point is modal. It gives motion. And that brings us up to the microscopic anatomy of skeletal muscles. Now, this can get very real very fast, folks, all right? Very real, very quickly. My goal is to make this as non-intense as is physically possible, but we're gonna see how this pans out and hope for the best. So with that in mind, let's get started. All right, here's how this works. Your skeletal muscles are unique. They're just unique. And because they are unique, you see some unique term sets applied to the parts and pieces of them. For example, anything sarco, you gotta be thinking muscle, okay? Especially skeletal muscle. Anything myo, you gotta be thinking muscle, okay? So let's go through and talk a little bit about these, what makes them special and unique, and then move on from there. Okay, skeletal muscle cells are big. Way larger than any other muscle or any other cell in your body. Okay, these are big, honking cells, very hefty, very long. There are longer cells in the body, but none that are as large and diameter as is a skeletal muscle cell. And as a result of being so big, they're multinucleated. Imagine you got a muscle cell yay long. You got to have multiple nuclei along the length of that thing to make the protein necessary to keep it working. Okay, and they are packed, and I mean packed with mitochondria as well. The outer covering of the muscle is referred to as being the sarcolemma. Now this is a cell membrane that is so different. Okay, it's just so different that we ascribe a different name to it. We call it the sarcolemma, and we'll talk about what makes it different here in a little bit. Muscle cells are like every other cell. They have cytoplasm, but the cytoplasm of a muscle cell is so different in chemical makeup that we don't call it cytoplasm, we call it sarcoplasm. All right, they're just different. These muscle cells, these skeletal muscle cells, are just different, and as a result of that, they get special names for their function. All right, now, the three main concepts about skeletal muscle that you have to be familiar with are: you need to know about the myofibrils. And this is complicated, team. You need to know about the sarcoplasmic reticulum. The sarcoplasmic reticulum is basically the smooth ER of a skeletal muscle cell, but it's so different. It's so different from every other cell in your body that we don't call it smooth ER anymore. We call it sarcoplasmic reticulum, okay? And then last but not least, we have Ttubules. These are parts of the cell membrane, and they are very unique, okay? Very unique compared to most other cells in the body. All right, now, myofibrils. When I say myofibrils, I'm talking about actin and myosin. Okay, the myofibrils are called actin and myosin. These are two protein sets, and their interaction, the interaction of actin and myosin, works to shorten muscle fibers. In other words, when I flex my muscle, I'm changing the distance between the origin and the insertion by having the actin and myosin molecules glide past one another like so. Actin and myosin don't change length. They just interact with one another and pull against each other like this. It's kind of like you can climb a ladder, but you don't get taller. Same concept, okay? The myofibrils don't change length. They simply move past one another. And when you look down here, you can see them. The red here, that would be myosin, and the blue here, that would be actin. These are myofibrils. And this results in the striated pattern that we see in skeletal muscle. I was going to take this a little deeper. Let's not go much deeper. What we have here is this striated pattern, repeating light and dark bands. We call these actin and myosin. The myosin bands are darker. The actin bands are lighter in color. Man, I feel like I'm skipping around here. All right. Now, we need to talk a little bit about neuromuscular junctions, and then we're going to get into how the muscle physically contracts, and that is complicated, so pay attention. All right, a neuromuscular junction, that is a junction between a neuron and a muscle. When I think flex bicep, I send a message down the length of an axon from my nervous system, down the length of an axon to my bicep. It stimulates that bicep, and the bicep contracts. Because there's a neuromuscular junction, also referred to as a synapse, there at the muscle. A junction between the nervous system and the muscle. Neuromuscular junction. Now, neuromuscular junctions have three parts. These three parts are the axon terminal. That will be the terminal end of an axon, the end that interacts with the muscle. A synaptic cleft. This is a little gap. Okay? A little gap between the axon terminal and the actual receptive part of the muscle fiber. That's called a synaptic cleft. And then last but not least is a motor end plate. The motor end plate is the receptive portion of the muscle fiber. You can see it here. What we have is an axon terminal. The axon would come down, 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 and you would end up with a terminal end of an axon. That is an axon terminal. Then you'd have a synaptic cleft. That's the gap between the two. And then last but not least, the receptive portion of the muscle fiber, it has special receptor proteins, gated ion channels, that are adapted here for increased surface area, and that is the motor end plate. So we got motor end plate, we got synaptic cleft, and we have neuromuscular, we have axon terminal. There we go. Axon terminal, synaptic cleft, motor end plate. Yeah, that'll do. Now, inside of this axon terminal, you find little vesicles. These vesicles are called synaptic vesicles, and they contain a very special chemical that we're going to call a neurotransmitter. That's probably on here someplace. Yeah, here we go, neurotransmitter. These neurotransmitters stored in vesicles are going to be allowed to release via exocytosis. They can cross the synaptic cleft and bind to this motor end plate and stimulate the muscle therein. In fact, that's exactly what happens. When I think flex bicep, a stimulus goes down the length of an axon to my axon terminal in my bicep. It releases a neurotransmitter that crosses the synaptic cleft, binds to the motor end plate, and the muscle flexes. Okay? As long as there is neurotransmitter in my synaptic cleft binding to my gated ion channels down here, that muscle is contracted. And when I decide to, I can cut that off and the muscle relaxes. Turn the neurotransmitter back on, muscle contracts. Turn it off, muscle relaxes. Okay, it's all about this axon terminal, or this axon coming down to an axon terminal, releasing neurotransmitters that bind to receptor proteins, bind to receptor proteins on the motor end plate, stimulating that muscle, leading to contraction. All right, I'm pretty happy with that. So the neurotransmitter is stored in synaptic vesicles. That's what they're called. What stimulates its release, we're not terribly worried about yet. They release by exocytosis. So exocytosis is how you get the synaptic vesicles to crack open. Yeah, all right, that'll work. Let's go here. Now, we need to talk about a couple other things before we can go further and put all this together. Those couple other things are the sarcoplasmic reticulum and the Ttubules. Okay? Sarcoplasmic reticulum and Ttubules. The sarcoplasmic reticulum is going to be this seen here in blue. Okay? All this blue stuff. Man, I wish you guys could see. This screen is going crazy. Anyway, neither here nor there. I hope the screen's not breaking. Moving on. The sarcoplasmic reticulum, all this blue here, in essence, is a big calcium storage bank. Okay, large calcium storage in the muscles. The sarcoplasmic reticulum has special modified proteins internally to store calcium. All of this blue, these are referred to as the terminal cisterns of the sarcoplasmic reticulum, the thick parts here and there and there. But that whole sarcoplasmic reticulum just stores copious quantities copious quantities of calcium. Okay, lots and lots of calcium are stored here, and they are in direct contact with the Ttubules. The Ttubules are our third modified structure. The Ttubules are seen here in purple, okay, in purple. And if you look at the sarcolemma, the outer covering of this muscle fiber, you can see tiny little holes, little bitty holes, holes all over the place, little holes. These are openings into Ttubules. Now, let's put this together a little bit. When we have a muscle fiber, that muscle fiber is going to be really big. You know at this stage that skeletal muscle fibers are much larger, way bigger than other cells in the body. So they struggle with diffusion. They struggle with carrying signals throughout their length and width, if you will. So if I've got a really big muscle fiber and I'm going to stimulate that thing, all I can do is stimulate the outside. I can stimulate it from the outside and hope for the best. The diffusion in would lead to the internal parts, then leading through contractions. But that would take a long time. The muscle's too big. If I stimulate the outside, I need to find a way to get that stimulus in deep to the core of that muscle fast. How? How can I get a stimulus from the outside of the cell to the inside quickly? I have Ttubules. All right? These Ttubules carry a stimulus from the outside all the way down to the core of that muscle fiber instantaneously. Okay? Instantaneously for our purposes. They carry a stimulus. Ttubules carry the stimulus from the outside of the cell to the inside of the cell quickly. And what are the Ttubules connected to? They're connected to the sarcoplasmic reticulum. What does the sarcoplasmic reticulum store? Calcium. Can you see where we're going here? If I have a stimulus coming down an axon that hits an axon terminal in my bicep and causes the release of a neurotransmitter that crosses the synaptic cleft and binds to the motor end plate there, what it will do is it will set off a stimulation that travels down the length of that muscle fiber. But let me try again. But does that stimulus stay on the surface of the muscle fiber? No, it doesn't. It travels down deep to the core of the muscle fiber using Ttubules, where it will then stimulate the sarcoplasmic reticulum, venting calcium, which tells the myofibrils it's time to contract, and you get contraction. It's the calcium release from the sarcoplasmic reticulum that triggers the actual muscle contraction internally. Okay, you probably have seen these dumb commercials where people buy like electrodes that you put on your abs or something supposed to shock yourself into having a six-pack, or you may have shocked yourself before and felt a twitch. Okay, what's happening there is you're cracking open these calcium channels and allowing calcium to interact with your myofibrils, and it leads to muscle stimulation. I could take a syringe full of an aqueous calcium solution and stick it in one of your muscles and you get a cramp like you've never felt before. The muscle doesn't care where the calcium comes from. If it gets calcium, it's going to contract. So let me walk you through this one more time, make sure we're on the same page. Let me rephrase, let me go backwards. If I come down an axon to an axon terminal, stimulate the axon terminal, it's going to release neurotransmitter, which crosses the synaptic cleft. It's released via exocytosis, crosses synaptic cleft, binds to special gated ion channels, which open, cause an outside stimulation of the muscle fiber. That stimulus travels down the length of that muscle fiber, but it doesn't stay on the surface. It travels down deep into the muscle fiber using Ttubules, which then stimulate the sarcoplasmic reticulum to vent calcium. That calcium interacts with the myofibrils, being actin and myosin, and leads to contraction. Okay, so let's talk about how this is done a little bit deeper. What you see is, imagine a stimulus, and that stimulus is flowing down the surface of that myofibril, or a muscle fiber, I should say. Stimulus is flowing down. It doesn't stay on the surface, it travels deep into the muscle fiber using the Ttubules. Can you see that? In via the Ttubules, where it would stimulate the sarcoplasmic reticulum to vent calcium, which interacts with the myofibrils, being actin and myosin, and leads to muscle flexion. Speaking of these myofibrils, The myofibrils are actin and myosin. We think about these as being the thin filaments for actin and the thick filaments for myosin. Now let's talk about myosin first. I think it's a little easier. Myosin is just myosin is just myosin. Myosin, the thick filaments, they're just big bundles of myosin. There's nothing terribly fancy going on with them. What makes them special, like hundreds of myosin molecules in a single big myosin piece of thick filament. What makes myosin special is it carries ATPase, meaning that myosin is what ATP works on to lead the muscle contraction. In fact, myosin has these little arms, basically, these little heads, or I think of them as like little grabby hands. And myosin will grab a hold of actin and pull against it, like actin is a ladder and myosin is tugging on that ladder. All right, do I have another picture of this? Hang on. Nope, I do not. All right, what will happen, again, just to make sure we're clear, myosin has ATPase, and it's got these little grabby hands. Not even close to this screen. What is it doing? It's got these little grabby hands, and it will grab a hold of actin, and using ATP, it will drag itself along the length of actin, leading to that muscle shortening. Okay, so myosin does the work. That's the idea here. Myosin does the work. Myosin has ATPase. So what's the role of actin? Actin is like the rope upon which myosin pulls. But actin has another very important nuance. Actin decides. Okay, actin decides if myosin can bind or not. It decides if there's going to be flexion. Actin decides. Myosin is always ready to grab a hold of actin and start pulling. Myosin is always stored at high energy. In fact, myosin stores up excess energy, so it's ready to go, man. Myosin is like, anytime you're ready, I will grab a hold and start pulling. But actin is not always available to myosin. In fact, it depends upon an ion influx. You want to guess which ion? What's stored in the sarcoplasmic reticulum again? Right, calcium. Okay, turns out that actin is complicated. In fact, we refer to it as being an actin complex. Actin is made up of actin subunits, seen here in blue, and then two more protein units called tropomyosin and troponin. Okay, troponin and tropomyosin. The way this works is that the actin is always ready to be bound to, but troponin and tropomyosin block the binding sites. Troponin kind of holds the strand like tropomyosin over the actin binding site so they are not free. You can see troponin here holding the strand of tropomyosin, blocking the actin binding sites. The only way to get troponin to pull tropomyosin out of the way and reveal actin's binding sites for flexion to occur is by giving troponin some calcium. If troponin comes into contact with calcium, it changes shape, frees up the actin binding sites from tropomyosin, and leads to muscle flexion. So you can see something yellow coming in here on this image. That yellow is calcium, and it's causing troponin to roll over and pull tropomyosin away, revealing actin's binding sites. So I think flex bicep, and a stimulus travels down my axon, down to my muscle, hits a neuromuscular junction where it stimulates the axon terminal to release a neurotransmitter. That neurotransmitter crosses the synaptic cleft, binds to the motorin plate there, stimulates the surface of the muscle cell, but that stimulus does not stay on the surface. No, it travels deep into the core of the muscle cell via the Ttubules, which then stimulate the sarcoplasmic reticulum. That sarcoplasmic reticulum releases calcium. That calcium binds to troponin, which pulls tropomyosin out of the way, revealing the actin binding sites, and myosin grabs and starts to pull. That's how muscles flex. All right, that's about the most complicated thing we're going to do this semester. That's how muscles flex. And just so we're on the same page here, the actin and myosin don't change length. They simply move past one another. Again, think about a ladder. When you climb a ladder, you don't physically get taller, you just move up something, okay? The ladder doesn't change length and you don't change length. The actin and myosin don't change length either. They're like my fingers and they just glide past one another. In fact, there's a term set for it. It's called the sliding filament model of muscle contraction. These filaments attract one another and simply move past each other, leading to the muscle shortening. And that's what I'm showing you down here. What you can see here are thick filaments and thin filaments, and they're simply moving closer or further apart from one another. All right, and then we must relax the muscle once it's flexed, and this is exactly the way you would expect things to work. Do I talk about neurotransmitters? No, I don't think I do. Let's not worry about it if we don't have to. I'll mention it very briefly. Okay, so what you'll do if you want to relax a muscle is, one, you stop stimulating it. So you stop releasing new neurotransmitters, and then you have enzymes in your body that break down the neurotransmitter that's still there. The side effect of that is that you stop stimulating the muscle fiber's length. So that means that the sarcoplasmic reticulum starts picking up all the excess calcium and putting it back into storage. If you store away all the excess calcium, that means that there's no calcium for troponin, so troponin rolls back over and blocks the actin binding sites. And if you block the actin binding sites, then myosin can't grab and pull, so the muscle relaxes. Okay, so you can think of muscle relaxation as being somewhat of the opposite of muscle flexion. Now, it takes a little longer for the muscle to relax because we're having to pump all these ions back where they go. We're having to use aerobic respiration to replace all of the ATP and such that we've used. If there's any damage, we got to fix that damage internally. So the act of relaxing a muscle takes a little longer than the act of flexing a muscle, but all the same, you know, it's sort of the reverse process. Now, we need to talk a little bit about muscle physiology. The first thing to mention here is the concepts of tension and load. Tension and load. Imagine my drink here weighs a pound. That would be a pound of load. If I'm going to hold my cup up, I need to generate a pound of tension. All right, if I generate more than a pound of tension, the cup rises. If I generate less than a pound of tension, it drops. The concept is tension and load. How do I generate more tension to overcome a certain amount of load? And that's what we're going to be dealing with here. And it revolves around this concept of the motor unit. Imagine this. Let me grab a chair. All right, here I've got a nice chair, okay? I can apply a little bit of tension to it, and I can kind of budge it off the ground a little, okay? I can apply a little more tension to it, and I can kind of stand up on it, or I can apply more tension to it, and I can lift it up, okay? So I can vary the amount of tension that I generate to overcome certain amounts of load. How do I do it? Can I make the whole muscle flex more weakly or more strongly? No. Muscles either flex or they don't flex. That's how this works. That's how your myofibrils function. They're either gonna do work or they're not gonna do work. They're either on or off. It is binary. So how do I make a muscle generate more tension or less tension? You do this by activating more or less of the muscle itself. All right, by activating more or less of the whole muscle by its lonesome. These are referred to as being motor units. Your muscles are broken down into individual parts called motor units. Whenever I grab this chair and I try to pick it up, when I budge it, I maybe activate 4 or 5 motor units. If I want to lift it up off the ground a little bit, I'm activating 15 or 20 motor units. If I want to lift it, I activate 50 or 60 motor units. The muscle is broken down into pieces. And by activating more or less of those pieces, I can generate more or less tension. When I get a hold of something really super heavy and I try to lift that up, I activate every motor unit I have simultaneously. And that's full muscle flexion, okay? That is how this is done. It's how this is done. If you look inside the muscle, you can see lots of individual neuromuscular junctions. The muscle will be broken down into individual motor units. Muscles that require great dexterity, like imagine your fingers, okay? Your fingers you must be very, very dexterous with to do the things that we do. So your fingers, the muscles in there have lots of motor units to give you a great deal of dexterity. Whereas the muscles in your legs, like your quads that just kind of hold your body up or not, they have very few motor units by comparison because they don't need that level of precision, okay? But the muscles are broken down into motor units. That is the main take-home message here. All right. Now, man, this thing is going crazy. Do you guys see all this weird color patterning? So strange. All right, let's start up here. Okay, let's talk about physiology of muscle fibers. Now, there are a few things to talk about here. The first we need to discuss is a single individual muscle cell and how it can twitch, a muscle twitch, a response of the motor unit to a single action potential. A single stimulus leads to what? And what we have here is time, and what we have on this side is tension generated. So how much tension does it generate over time? How strong is it over time based off of one stimulation? I could take an electrode, put it on your bicep, and give you a zap, okay? And when I give you that single zap, you'll see the arm twitch and relax. And that's what we're recording here, okay? What you'll notice is there's a little bit of a latent period, a little bit of lag, okay? Just the most brief moment where nothing happens. That's where you've zapped the muscle and it's starting to vent calcium. The calcium binding to troponin, and then it's moving tropomyosin out of the way, and then the muscle can finally start to contract. Okay, so there's a little latent period where the muscle is like gearing up for contraction, and that's followed by that moment of contraction. Okay, you get this peak of contraction, and then it begins to tail off slowly. Why does the period of contraction take less time than the period of relaxation? Because it's a lot easier to let these ions out than it is to pack them back into where they're supposed to be. They go out via diffusion, quickly, very fast, whereas you have to put them back where they go via active transport. Okay, you must actively transport them back to where they are supposed to be. And what you'll see is different muscles twitch at different speeds. Your eye muscles are very fast. I think you would expect that, like they can move very quickly, whereas like your leg muscles are very slow by comparison. And one of the reasons for this is the faster a muscle contracts, the faster it can fatigue. So you'd expect your leg muscles, which you have to, stand up all day long, to be capable of working for long periods without fatiguing. So they have a very slow, what we call ATPase. The ATPase, the enzyme that breaks down ATP, it does so very slowly for their meiosis. Okay. And then we need to talk a little bit about graded muscle responses. How deep am I going to take this? Let me see. Yeah, okay. So think about this as being a single muscle fiber, and when it's stimulated, how you can make it generate more tension. So I can send a single twitch, to a muscle, a single twitch, and not much happens. I'm not lifting a chair using that single twitch, okay? So here we have our twitch, bam, poof. You get this little up and down. Well, you get this, come on machine. You get this little up and down. That is that period of contraction followed by a period of relaxation. You don't generate a whole lot of tension. That's tension over time. Where this gets interesting, team, is in what we call wave summation. Okay, wave summation. So that when we send a stimulus, if we immediately follow that by a second stimulation, it'll build off the first one and you'll get more tension generated. In other words, if I start sending several stimuli back to back to back, I can start generating more and more and more and more and more tension. And if I send lots of stimulation very quickly indeed to a single muscle, that muscle will build tension to a point that it maxes out and can go no further. Now, your muscles are only capable of generating so much tension. You know this. Like, we can bring everybody listening here into a room, line up barbells from 5 pounds to 500 pounds, and somewhere along that length, you'd reach a point where you can go no further. Your muscles are only so strong. So, doesn't matter how many stimulations you send, at some stage they max out. Okay, they max out. But this is referred to as being wave summation. A single muscle fiber can twitch, but if you send several stimuli back to back to back, you can generate more and more and more tension over time. Now, how do I get a muscle to contract more strongly as a whole? Well, we talked about motor units, yes? The idea is that I can, again, here's my chair. I can stimulate just a few motor units, or I can stimulate more motor units, or I can stimulate lots of motor units simultaneously. This is referred to as being recruitment. Okay, recruitment. The idea is I can stimulate a few motor units and generate just a little bit of tension. I can stimulate more motor units and generate more tension. I can stimulate lots of motor units and generate lots of tension, or I can stimulate a pile of motor units and generate maximal tension. So I can stimulate them all, in other words, and it'll go no further. Let me check something. Yeah, that'll do. That is recruitment. You're recruiting more motor units. I have a point to make, though. This is fascinating to me. If I grab my chair, I could lift it up and I could hold it there for probably, if I wanted to, I could hold it for a couple minutes, maybe 3, but it would start hurting so bad it would just be terrible. Okay, just absolutely terrible. Whereas I could sit here with my drink and hold it like this for the rest of this lecture, no problem. No problem at all. It would get irritating, but I could do it without any question. And what I would like to know is why that is. Why is it if I'm holding something light that I can hold it up pretty much forever, whereas if I'm holding something heavy, I can't do so. Can't hold it up for long at all. And the answer here is pretty fascinating. If I'm holding up something heavy, the whole muscle is flexing simultaneously. But if I'm holding up something light, only a few motor units are active. Guess what happens when those few motor units become fatigued? Whenever they've been used to the point that they're no longer capable of flexion, they cut off. And other ones cut on. And then those are used until they fatigue, and then other ones cut on, and then other ones, and other ones, and other ones, and other ones. So you can hold up lightweight things for prolonged periods of time because different motor units are taking the load. All right? Whereas if you try to hold up something up that's quite heavy, the whole muscle is going to be flexing, or lots of it is, so you don't have the ability to shut those fibers down and or turn on others. It's a pretty neat concept. All right, let's go here. Muscle tone. So muscle tone is a constant, slightly contracted state of muscles. This is done for a variety of reasons. Now your spinal cord will do this for you. This is not something that you are conscious of. You can't control it. Several days ago, I was doing some work in my garage, and I was having to hold on tightly to something while I was working on it. And when I was done holding on to it, I couldn't really relax my hand. Okay, my hand was in a state of tonus. It was mildly contracted. Because your body's like, oh wow, you're doing this activity, we better stay ready if you're going to do it some more, so we're going to send some mild signals to that muscle and keep it fresh and ready to roll. Now, what if you work out every day? If you're going to the gym constantly and you're getting lots of exercise, you can make a whole body state of tonus. You've seen it before. You ever go to the gym, you see guys walking in having a good time, but then when they walk out like this. Is that reality, or is it in their heads? I would say a little bit of both. You certainly feel like a big, strong person walking out of the gymnasium. But if they've been exercising, then their muscles will most certainly be in a mild state of tonus. And what this does is it keeps the muscles firm and healthy and ready to do work. And it helps to stabilize your joints. That's why we do physical therapy. A lot of the reason we do physical therapy is to help to stabilize joints and help hold things together. Absolutely. Let's go here. All right, let's talk about how we get ATP for our muscles. Now, there are actually 3 ways. 3 ways that our musculature gets ATP. This is through pathways we call the creatine phosphate pathway, aerobic cellular respiration, and what we'll call anaerobic fermentation. Okay, this is a little complicated. It's glycolysis, but fermentation all the same. I will say creatine phosphate pathway, aerobic respiration, and fermentation. All right, let's see here. Key point to be made. Yeah, let's just talk about it from here. Feel free to read this. I'll just tell you what happens. You actually store very little ATP. Okay, you store very precious little ATP. Just enough for a few seconds, really. I mean, maybe 5 or 10 seconds. You store enough ATP to stand up and run out of this room, but by the time you get to the door, you're going to be out of ATP. It's going to be done and over with. But if I had a probe in your musculature and I could measure the amount of ATP that's found there, there would be 0 measurable change in ATP. So if you measured the amount of ATP in my bicep and I flex and then relax, the amount of ATP in my bicep would be unchanged. You could not measure a difference because your muscles contain large amounts of creatine phosphate. Your muscles contain large amounts of creatine phosphate. Creatine phosphate, the moment ATP breaks down, it rebuilds ATP. ATP breaks down, it rebuilds it. ATP breaks down, it rebuilds it. You store enough creatine phosphate to keep the muscles working for 15 seconds, somewhere in that neighborhood. I mean, completely breaking down the ATP and rebuilding it immediately for about 15 seconds using the creatine phosphate pathway. So What if you need to go longer, 20, 30, 40 seconds, minutes? What if you need more ATP? Well, you normally do that through aerobic cellular respiration. Aerobic respiration is using oxygen, that's the key point here, using oxygen with your mitochondria, using sugars to build copious quantities of ATP. All right, the idea behind aerobic cellular respiration is we use our mitochondria, we use oxygen, and we make huge amounts of ATP. 30 plus, lots of ATP molecules per glucose. This is the efficient and effective best way to do business. Now, what if you can't keep up with your ATP demands using aerobic cellular respiration? I would invite you to pause this video, type into YouTube, and say, people collapsing at finish line of marathon. And you'll see folks that have run a marathon using aerobic cellular respiration. They are keeping up with their ATP demands using aerobic cellular respiration. And then at the end, they try to spread it out, and they're burning all the ATP they have, and they run through fermentative processes to build an emergency extra quantity of ATP. But the side effect of fermentation is that it releases things like lactic acid and other problematic compounds that cause issues for your muscles. And before those folks cross the finish line, they collapse with cramps. Because of this. You start running, 10, 15 seconds in, you're out of creatine phosphate. You're going to be using aerobic cellular respiration to make all the ATP you need as long as you keep a pace. As long as you can keep up with your ATP demands using aerobic cellular respiration, it's going to work like a charm. But then if you decide to sprint it out at the end, to really exert all the power you physically have, it ain't going to last very long. And you're going to end up with cramping and problems. That's the side effect of fermentation. Fermentation is going to be when you make an emergency last-ditch effort to make a little bit more ATP, and it's going to result in muscle fatigue quite quickly. All right? And we can apply term sets to these. There's aerobic endurance. Aerobic endurance is the duration of time you can maintain aerobic cellular respiration and keep the muscles flexing in that fashion. If you need an emergency supply of ATP, you might have crossed your anaerobic threshold. Anaerobic means no oxygen. In other words, this is fermentation. You're going anaerobic. If you're using all the oxygen you got and you still need more ATP, you'll start to do fermentation. That's anaerobic threshold. You cross the anaerobic threshold, and that can lead to muscle fatigue. You can injure the muscles, you can damage their sarcoplasmic reticulums, you can run them out of ATP, and all of this is going to run into cramping. Okay. Now, let me ask you a question. Does she immediately stop breathing after she runs? If you run a marathon, do you immediately stop breathing, like hard? Because you're like, oh, I'm dying. I crossed the finish line and I'm done. Finished. That was a nice run. No, you keep breathing hard for a prolonged period of time because you have to clean out the muscles of all the materials that have been released there. You got to rebuild your creatine phosphate storages. You got to get rid of any lactic acid buildup. There's a lot of work to be done by the body, so you keep breathing hard for a period of time so that you can get in copious amounts of oxygen and keep the system working the way it's supposed to work. Yeah, that'll do. Let's go here. Let's not delve into minutia. All right, muscle fibers also come in two basic types. The two types of muscle fibers are fast twitch fibers and slow twitch fibers. Fast twitch fibers are also referred to as being white fibers or white muscle, and then slow twitch fibers are thought of as being red muscle. Okay, you may have heard, like when you're dealing with chicken, you got white meat and dark meat. The idea here is that these are different types of muscle fiber. The best way I can see this is when you look at salmon. Do I have a better picture? I don't have a better picture. That's a shame. When you look at salmon, I don't know if you'll be able to see this image, but there's like this pink musculature on the outside. It's very light in color. And then right there, there's this really dark musculature. And they're different. They're very different. What that is fast twitch muscle making up the majority, and then a little bit of slow twitch muscle here on the sides of the fish. That's actually the swimming muscle. And then this bit here, the fast twitch, is like their emergency escape muscle. All right, let me explain how this works. Why would a muscle fiber appear light in color? And they do. They're quite light in color. The answer is very simple. They don't get a lot of blood flow. You have fast twitch muscle fibers. Fast twitch muscle fibers rely on creatine phosphate and fermentative pathways to make their ATP. As a side effect of this, they just don't have a lot of blood flow to them, so they have a light coloration. They are adapted for great strength. They're light in color. They have very few mitochondria. They have very little what's called myoglobin. It's an oxygen storage. They have very few blood vessels. Fast twitch fibers are the ones that are very big and strong, and when you pick something up, they're very strong, and then they fatigue quickly, and you gotta set it down, okay? Fast twitch fibers have burst strength. Very powerful, but for short duration. Think about this like a power lifter, okay? Like a power lifter. This guy here, his name's Thor Bjornsson. He's like been world's strongest man a bunch of times. Like he's a big, strong guy. And he's huge, like his muscles are massive. And he has mostly fast twitch fibers, so he could do great feats of strength, but then his muscles need rest for a period of time. They lack endurance, you might say. They lack endurance. By comparison, there is slow twitch fiber. Slow twitch fiber relies upon aerobic respiration. You gotta have lots of oxygen. And lots of oxygen means lots of blood flow. So because this musculature, slow twitch fiber gets lots of blood flow, what color is it gonna be? It's gonna be kind of red and dark in coloration. Okay? Adapted for endurance, dark in color, piles of mitochondria, lots of myoglobin, lots and lots of blood flow. This musculature is going to have really good endurance. Really good endurance. But the side effect of this is that slow twitch fibers, though they are wonderful for endurance, they aren't terribly strong. In other words, they're not very big. Fast twitch fibers, there you go. Fast twitch fibers result in really big muscles that are very strong but fatigue quickly. Slow twitch fibers result in muscles that are very capable of flexing over and over and over again. They have a lot of endurance. They're not as big, so they're not as strong. That is endurance exercise based versus powerlifting. And we can see it. We can see it. Look at Thor Bjornsson. Look how big his arms are. And then look at Andy Kaprotic, man. One of the greatest marathon runners of all time. Look at his legs, look at his arms. If you saw him, you'd be like, wow, he's in really good shape. But you wouldn't think he was a super hardcore Olympian because his muscles aren't that big. Because his exercise regime leads itself towards slow twitch fiber. Okay, towards slow twitch fiber. Endurance exercise versus powerlifting. Most of us, okay, this is genetic, man. Okay, that's genetic. These guys are genetically predisposed towards their exercise regimes. Most of us are somewhere in the middle. I don't know if I can say that Usain Bolt is average, but he's somewhere in the middle with the rest of us. Most of us have somewhat of a combination of slow twitch and fast twitch, as opposed to being a predominance of one or the other. All right? Slow twitch versus fast twitch fibers. And we can drive this through our exercise regimes also. So this is Michael Phelps. Michael Phelps is a swimmer. He does primarily aerobic-based exercise. And if you look at Phelps, you think he's in good shape, but look at his tiny little arms. You can guarantee that he has mostly slow twitch fiber as a result of aerobic or endurance-based exercises. When you do aerobic exercises, you're increasing the amount of capillaries around your muscles, you're increasing the number of mitochondria, you're making the muscles more efficient, but the muscles don't get bigger. You don't promote what's called hypertrophy, the enlargement of the muscles. By comparison, here's Arnold. Schwarzenegger himself, obviously on steroids at this time, but you get the point. By doing heavy weight, low reps, you increase the individual muscle size. The size of the fibers gets larger. You increase the tendency for them to be white muscle, this light in coloration, fast twitch stuff, and then you sort of augment it away from aerobic respiration. And that's good enough for me. So again, point being that based off of your exercise regime, you can vary the musculature which you express. All right, and now we got a few other nuances to talk about, and we're going to be real close to the end. So first is DOMS, that'd be delayed onset muscle soreness, DOMS. This is a situation where you can cause minor damage to your muscles as a result of doing some sort of exercise that you're not used to, or what have you. You can do physical exertion that you haven't done in a while, and it can cause minor tearing and damage to the muscles. Like, you're not going to hurt much. These are microscopic level, like tearing off some myosin heads. but it's going to lead to inflammation and pain, so you're going to hurt the next day. The idea with delayed onset muscle soreness is you hurt the next day. Okay, you hurt the next day. Let me check my clock here. All right, you hurt the next day. That's delayed onset muscle soreness. Compared to strange pulls and tears, this is when you hurt immediately. This is when your body tells you you're making a mistake. That's what pain is. Pain is your body saying, hey, stop screwing up. This is a bad idea. My genius brother, man, let me tell you, he was 10 years out of high school, and a buddy has called him up, and he was like, hey, come play church league softball with us. So we went out and started playing, and he thought he was just going to play ball like he did back in the day. He's throwing this heavy softball around. He's like, man, my arm's starting to hurt. So I look at it, and it's got this ring of blood around the bicep where he has torn it into two parts. This is a very traditional bicep tear. This is what it looks like, where you've literally torn the muscle away from its origin point, so it just gathers up at its insertion when you flex. If you are doing some sort of activity and you start to get pain, you should stop, because this is hard to fix, okay? You need to be very, very careful with it. So be careful is what I'm telling you. If you start to hurt while you're doing some sort of physical exercise, you need to stop immediately so you don't have permanent damage. Let's talk a little bit about hernias. How much do we want to talk about hernias? Not much. All right. A hernia is a situation where there's some sort of weakness in the abdominal wall and parts of the intestine can push their way through and not stay where they're supposed to stay. This is a classic umbilical hernia here, where the intestines have pushed their way out of the umbilical area. So you can see this quite clearly. This is confirmed via imaging techniques. Now, this can be caused by a whole host of things. Heavy lifting can certainly drive this. Bad posture leading to weird weaknesses in the abdominal wall can cause it. Car wrecks, you see a lot of folks get hernias after car wrecks. In any high pressure situations, believe it or not, there's a tendency towards having hernias for people with chronic constipation. So when you have this constant abdominal pressure, you can certainly force things where they're not supposed to be. This is easily noticed by bulges in the skin, and then again, we use imaging to figure it out. This tends to be more of a male thing. The male abdominal wall has inset weaknesses as a result of the positioning of sex organs, and it results in dudes getting hernias far more often than do females, far more often. So this is mostly a male thing, or predominantly, we'll say predominantly a male thing. All right, muscle compartments, oh, and the young. So most babies will have abdominal, umbilical hernias, just the nature of the beast, and they tend to go away over time as the abdominal wall gets stronger. Muscle compartments. So if you look at areas where there's a lot of muscle in a small part of the body that move past one another, like in the leg or the arm, so like in your arm you have like 6 muscles that cross the arm right here, and they're all just in parallel to one another. What you will do is you'll have individual little fibrous compartments that surround these muscles and keep them separate from one another so they can contract and do what they do without leading to friction-based damage. We don't want to have friction-based damage. Yeah, that's how this works. So each muscle gets its own blood supply, gets its own nervous tissue. You have these muscle compartments. But this can lead to problems. And the problem it leads to is referred to as compartment syndrome, and it's on here someplace, I'm sure. Do you see compartment syndrome anywhere on here? Anyway, somewhere in there, I'm sure it says compartment syndrome. Compartment syndrome can be very, very dangerous indeed. This is a situation where after like a car wreck or some sort of other mechanical impact, I've heard of people falling out of trees and getting compartment syndrome. You can damage a muscle inside of its muscular compartment, and it does what any damaged muscle does. It bleeds. Okay, it'll start bleeding internally. Maybe it's the next slide. Nope. It'll bleed internally. And as it bleeds internally, it will cause the muscle compartment to swell and balloon, and man, it gets solid like a rock, really hard. And you can see it too. Like I've seen where they've had like a stripe down the arm, a big red stripe. You can see through the skin and see like this spot where there's like this hard, weird thing happening. And that's compartment syndrome. And man, you got to be real careful with that kind of stuff. Because when you have that much pressure build up inside of a muscle compartment, the body can't pump blood into it anymore. And if you can't pump in the blood, guess what? No oxygen. No oxygen means necrosis. The tissue begins to die. And if the tissue dies, nerve damage, eventually muscle damage, it's not coming back. You don't heal that kind of thing. So you lose it completely. Really dangerous. The ways in which we diagnose this is our P's: pressure, pallor, pulse, and paralysis. Pressure, if it feels hard. Pallor, if the color is weird. Pulse, if you can't get a pulse from an artery in the area, probably means there's no blood flow to it. And then paralysis, it ceases to work. What we do in this particular situation is called a fasciotomy. Fasciotomy, I'm sure I've got it on here. Fasciotomy. A fasciotomy is when you go and you cut the fascia. Imagine if I've got compartment syndrome and a muscle compartment here, I can go in and cut that compartment open. And I've never done it, but I've seen it. And I've heard from my friends that do paramedic stuff, whatever you call that. Man, it's quite a show. Okay, it's a bloody mess. When you crack open that water balloon of blood, man, it's coming out. Okay, but by relieving the pressure, you allow blood flow back into the area, and you can get healing. I had a former student, and she came up after a lecture like this. She said, "Hey, I want to show you something." Pulls her sleeve up, and pretty much she just has the bones. Like the hand is not really working, and all of this, like all of that is just gone. It's flat. It just looks like she's got like the bones there, man. And I was like, "What happened?" She goes, "I got in a car wreck." And went to the emergency room. I had a big bruise on my arm. And I was like, "Yeah, I can't run my hand." They're like, "Yeah, you know, it's going to be like that for a few days. Once the swelling goes down, you'll be fine." And she had compartment syndrome. They didn't diagnose it correctly. And she lost all the musculature in her arm. And I mean all of it. It was gone. It was the strangest looking thing you've ever seen. Terrifying, man. Absolutely terrifying. So be vigilant. Be vigilant. And be careful. Yeah. And once that happens, the muscles start to vent off compounds that lead to kidney failure too. So that could kill you real fast. All right, rigor mortis, snake venom. Tell you what, team, I'm pretty satisfied. I think we're done. I think it'll do it. So let's stop it here, and you'll have a quiz up real soon. Test upcoming, not terribly long from now. Have a good day. Enjoy. Thank you.